As a refresher from our last video, stem cells are different from any other cell type in the body. They have three unique properties. They are able to divide and renew themselves for long periods of time, they're unspecialized, and they give rise to specialized cell types throughout the body. Unlike cells such as muscle cells, nerve cells, or blood cells that don't normally replicate themselves, stem cells have the ability to replicate themselves many times over. A starting population of stem cells that proliferates in the laboratory for many months has the ability to yield millions of cells. Scientists are trying to understand the two fundamental properties of stem cells that relate to their long-term self-renewal. The first is why most embryonic stem cells can proliferate for a year or more without differentiating, but most non-embryonic stem cells cannot. Secondly, what are the factors in living organisms that regulate the proliferation of stem cells and self-renewal? Once scientists discover the answer to these questions, it may be possible to understand how cell proliferation is regulated during embryonic development or how abnormal cell division can lead to cancer. The conditions and factors that allow stem cells to remain unspecialized is very important to scientists. This information is critical in order to grow large numbers of unspecialized stem cells in the laboratory in order for further experimentation. The most important property of stem cells is that it does not have tissue-specific structures that allow it to perform special functions. For example, a stem cell cannot pump blood through the body like a heart muscle cell, or carry oxygen through the bloodstream like a red blood cell. Stem cells can, however, give rise to the specialized cells. When unspecialized stem cells give rise to specialized cells, this process is called differentiation. The differentiation process has many stages, allowing the cell to become more specialized through each stage. Scientists are just beginning to understand this process, and the signals inside and outside of the cell that trigger each stem of the differentiation process. The internal signals are controlled by a cell's genes, which are interspersed upon long strands of DNA and carry coded instructions for the cellular structure. The external signals for cells include chemicals secreted by other cells, physical contact with neighboring cells, and molecules found in the microenvironment. The cell's interaction of signals during the differentiation process causes DNA to acquire epigenetic marks. These marks can restrict DNA expression and be passed on through cell division. There are still many questions remaining about the cell differentiation process. For instance, are the internal and external signals present during differentiation the same for all types of stem cells? Also, can specific sets of signals be identified that promote differentiation in specific cell types? Finding the answers to these questions can lead scientists to find new ways to control stem cell differentiation. This will allow them to grow cells or tissues that can be used for purposes such as cell-based therapy or drug screenings. Tune in for a future video where we'll discuss embryonic stem cells, adult stem cells, and the difference between. Thanks for watching my video on the unique properties of stem cells. Please rate my video, leave whatever comments you would like, and subscribe to our channel.